So I've sent Brian an email asking him to keep his voice down. I think where he is now is okay. But keep an ear out. If he like really starts spouting, I'll need you to go to his window and do like one of those kind of things. Thank you.
I'm gonna sit in here. And I won't leave it all the way closed, but I can kind of see it through the window.
Okay. Welcome to those of you who are starting to log in for today's webinar. We will get started with Dr. Robert Gallo at the top of the hour. Welcome to those of you who are signing in for today's webinar with Dr. Robert Gallo. We will be starting at the top of the hour. So thanks for logging in. Welcome to those of you who are logging in for today's webinar with Dr. Robert Gallo. We will be getting started at the top of the hour. Hi, everyone, and welcome to today's webinar focused on the COVID-19 pandemic. I'm Chris Rebelo, the Director of Communications here at the Buck Institute for Research on Aging. This is our second series of webinars focused on the fight against COVID-19. I have the honor of interviewing many experts in the field during this series. Next month, we'll be talking to the current director of the Center for Disease Control. Today, we talk with one of the most honored biomedical researchers in the world. Dr. Robert Gallo is best known for co-identifying HIV as the virus that causes AIDS. He is currently the co-founder and director of the Institute of Human Virology at the University of Maryland School of Medicine. Dr. Gallo was the most cited scientist in the world during the 1980s. He is a member of the National Academies of Science and has won the Albert Lasker Award for Medicine twice. He's also the co-founder and international scientific advisor of the Global Virus Network. Although Dr. Gallo has devoted much of his career to the fight against AIDS, he has pivoted to the fight against COVID-19. He is spearheading a project that involves the polio vaccine. We will be talking to him about that. And as always, a reminder that this is an interactive event. If you have questions for Dr. Gallo, please use the chat feature at the bottom of your screen. I'll try to get it to as many of your questions as possible. So welcome, Dr. Gallo. Thanks so much for making the time to be with us today.
Thank you for having me. It's a pleasure.
My pleasure. So I always start these things off. I'm in California, you're in Maryland. How's it going there?
I really don't know. I don't follow day to day. It's, it's not so great in a month preceding, 2 months preceding, but a bit better now. The county I live in, surprisingly, is one of the worst, if not the worst, Montgomery County, Bethesda, right in the outskirts of NIH, et cetera. So I don't even know why it's so much here, but it's, the statistics show it's getting better. So what can I say? But is it great? No. And we get hit very hard with the nursing home, that's for sure. So that's what I can tell you. But I don't like, I know we have to. I mean, somebody has to follow the numbers. I don't, personally, I don't enjoy following numbers so much. We got a problem. We have to solve the problem. I'm a little bit obsessive that way. What I focus on, I want to stay with. But if you start following the numbers all over the place, you get so you think you can predict. Like I heard yesterday from our friend Tony, that there are some states going up. Yes, we know. And in 5 weeks, we'll know if it's going to be better or not. Well, how about in 1 week, or how about in 10 weeks? How do you know in 5 weeks? And better spend your time on thinking, what are you going to do about it? I mean, you know what I'm trying to say.
Yes, I do. So that's a good segue. Just for numbers, though, I will say, I read in the New York Times today that they said that there were 35,000 new cases yesterday identified in this country, which brings me to the project that you're involved in, which is taking a look at the Oriole oral polio vaccine. So talk to me, what's that about?
I thought you were going to start talking about the Baltimore Orioles, and they're not that good, in these days. So I said, oh God, she's going to get into that. What do I do? Sure, I'm going to talk to you about it. You want to begin with the idea?
Sure. Talk to what are you?
Why did I get into it?
Yes.
Yeah. Oh no, we'll definitely get into what I think it'll do. Hope it'll do. Believe strongly it will do, if we can get it going. The idea is not new. We like to take a certain pride. We have new ideas. It's not original, though. By genetics, there's a relationship, because my closest colleague in this is Konstantin Shumakov, whose father's picture is in, among others, in the hallway that as you enter our institute. He's one of the great virologists ever. He was Russian. Shumakov came to America as a young man and became associate director for vaccines at FDA. It is his mother, working with his father, who made the first critical observation. Albert Sabin of this oral polio vaccine, who made it, who I used to know quite well, as well as Salk, I kind of bridged that generation, has said in writing that the whole vaccine concept wouldn't have gotten anywhere without Shumakov. He's the one who did it in Russia and showed it worked. That was 1950s early, so it's 70 years ago. It's got a history of 70-year use, long time. And then about 20 years later, his wife, sorry to say, I'm as bad as they were then. She has her own name that she used in science, but as you know, women weren't always remembered that well, but it starts with a V. I remember that. She made the observation that, wow, the same year they were vaccinating, they didn't see the influenza epidemic that was coming. And there was a 3.8-fold diminution. And that's a real wow. That was better than the influenza vaccine. So they knew there was some kind of heterologous protection. And she coined the term virus interference. And a few years later, interferon was discovered. I suspect, do not know, that it was given that name based on what she coined that term. And indeed, the innate immune system, which we'll talk about, is highly dependent on one of its key components being interferon, and it's part of what we'll come back to and talk about. So they reported that, and it got reported again in Russia and then in Singapore. Then the oral measles vaccine did the same thing with other bugs and would seem to be broadly protective against a wide range of things. Slow realization in time of the science that the RNA sensors in our cells were picking up foreign RNA molecules and instigating this immediate response within a few hours, not like the adaptive immune system. And it's an emergency response, as you can imagine, and as you know. It's a hot area of basic immunology today, one that I do not regard myself as anywhere near expert in, but I'm trying hard to learn as much as I can. As I said, a key component is interferon, the myeloid cells like monocytes, macrophages, but there's a lot more to it. And this is a chance to learn a lot more and be applicable for future potential epidemics and pandemics, as well as future waves of this.
Yeah,
but I want to finish this thought.
Let me ask you first. So is there a clinical trial that's going to look at this vaccine for COVID-19?
Yeah, of course. But you got to tell me, how much time do I have? Because I was certainly going to go there.
Oh, we have about 50 minutes left, but there's a lot I want to talk to you about.
Okay. But of course, I'll get to that. But I did want to say that our point, it's also, we're not resurrecting a dead concept also. It's not a Lazarus phenomenon to those who know the story of Lazarus, but rather you might think of it as a premature infant that we're trying to develop. Okay, something like that. So we're right. So the idea is important to know how it comes about. We were having a global virus network discussion. That's 55 centers in about 35 countries. And we influenced Chumikov to be, he's, FDA, though FDA, he's got a lab that's part of the GVN. And I remembered his discussion with me a long time ago about this. And so I raised the question, nobody paid any attention to it. Then I thought, Jesus, this is important. So I had a meeting with him after. The idea developed further, and we can talk about what that data is. But if you want to jump from that all the way to the clinical trial, we are involved as consulting collaborators with trials that are beginning in several different countries right now. And they started, some just starting, some already operational for several weeks. We began these discussions in the beginning of February. It's a long time. It's February, March, April, May, June. And we got a lot of support to begin a trial in the United States. And it was with excitement by your next speaker next week, who, by the way, also co-founded the Institute of Human Virology with me. And also by the director, our director who's on the television every day at NIH, the One Institute, very enthusiastic, his associate, very enthusiastic, and 14 people from NIH made a site visit, very enthusiastic, and then all of a sudden stopped. They're going to evaluate all the spike vaccines first. There doesn't seem to be a plan B. Everything is the spike and neutralizing antibodies. Dude, that's obvious and simple. You have the sequence from the Chinese in January 10th. You can sit in your basement if you have enough money and order it. You get the message, put it in the right solution, and you inject it, and you'll get the spike protein and you'll make antibodies. There are some reasons to be a little skeptical that you might want to have a plan B rather than 5 spikes going forward. I won't get into all of that unless you ask me, but I'll tell you what makes me, wary of not having a plan B. Yes, this may work. Yes, they should go forward, but I believe strongly there should be a plan B. Meanwhile, this could have been out months ago. So the clinical trial that we had involved some important people for studying the innate immune system, as NIH wanted and as they were thrilled by. The Cleveland Clinic, it involves other people that are pros in that. And it involves several centers where we'd have enough patient population. And this was set to go. Now it's on hold here. We have another source of funding to do something different. We're going to, in a collaboration with European people, we're going to deliberately infect people, not with SARS-2, but with the seasonal coronaviruses that cause common cold, and separately with influenza. We're going to get answers fast, and we're going to hopefully learn something about the mechanisms that correlate with protection within weeks. When does that start? Well, that is developing now, funding-wise. This week, next week, we'll have all the answers to that. And right now, it's very positive. So I think that'll happen. And the things in different countries will happen. Includes about a half dozen different countries, maybe more now. And we are kind of pushing this still for the trial in the United States, sponsored by where we hope to be sponsored from NIH. There's no way that this is something we're giving, slow down on, that's for sure.
So when you mentioned Plan B, this vaccine could be a Plan B. You hope that it would buy time until the.
Yeah,
What would it do?
first of all, I mean, look, this is completely safe. So there's been these ruses. I don't know what the right word is. When you throw something in the air, you don't really know what you're fully talking about. Oh, it might get in the water supply. Oh, it might do this or it might do that. We can come back to safety in detail in a moment. Oh, are we sure about efficacy? Are you sure about efficacy with the adaptive vaccines with the spike? But there is a ton of data that this works. I mean, a lot of data. Okay, yes, a lot of it came from Russia and elsewhere, but we're just ignoring it. It works. And so if you can give it again and again and again, and you may extend the innate immune response, the emergency response, let's say the first responders. You may extend it for several months. We expect 2 months with one, maybe a little less, maybe a little more. With BCG, by mechanisms they don't understand, called molecular imprinting, they're getting out to a year. This should be far stronger in its immune stimulation and much simpler. It's a drop on your tongue. And Morris Hilleman was a great vaccinologist at Merck. He gave us the vaccines that you and I take, with the exception of rabies and polio. He was on my board for years, and he used to always say, don't even begin a vaccine unless you know feasibility. Feasibility means I don't know if it's, if it's going to be effective, but it damn well better be inexpensive, easy to give, survive cold chain, have a history, know what you're doing, that it can be done. Nothing is better than the oral polio vaccine for that purpose, with experience. If you want to go into safety, tell me when, and I'll be glad to tell you what data I'm aware of after having to be aware, to know such data.
So talk about safety. And also, I think when we talked earlier, you said it could be a way to kind of get people back to work, get things moving again, right?
Yes, I'm writing something about that right now, Chris. I mean, I hope it gets published. I think it will, but you never know. But that's something I want to really, as medical scientists, we don't think so much about that. But when somebody really tells you the story, if the economy continues to collapse, people suffer in other ways and get sick for other reasons that you're not thinking about as medical scientists. You've got to think about the economy and people getting back to work. So sure, my purposes are: 1, getting people back to work; 2, having an immediate prevention of death, which we could have had months ago; 3, what about the fall season with influenza? And if a new guy comes back resistant to the vaccine, if the vaccine works, if it's safe, if it's durable, which I have my doubts about. So you would love to have something like this. And how about for a new pandemic? And let's say we learn something about the mechanism with real pros that study innate immune system. Wouldn't that be great? Wouldn't that be really valuable? To turn to safety, let's start with all Americans between 1962 and 1997, for those 37 years, took oral polio. Little drop on your tongue. In those days, it was a sugar pill. Many of you listening to this might, well, some of you anyway, probably remember this. It was pretty darn safe. Billions, not millions of people have received oral polio vaccine by now. The safety record in unvaccinated people is about 1 per 2 million. A complication about 1 per 2 million. That's pretty safe. But if you're in a vaccinated population, to the best of my knowledge, and double checking with Chumakov, doesn't know of a single case of anything.
Hmm.
And look at all the disease savings, and look at here. Look at older people, me. I mean, I don't want to go back to work until I have an oral polio in my mouth. Okay, so that's the way I see it. And then the argument comes, oh, it might get in the water supply. I mean, that's when I have to bite my tongue hard, because I tend to not always be in total control of my emotion. And I say, well, it's in the water supply. What survives in the United States water supply? For the last 150 years, I don't think any virus has survived, but let's take it at face value. Israelis had oral polio, 6.5 million of them, not so long ago. And Chinese, Pakistanis, Indians, many people all over the world. Do we not go to those countries? Do we block them from coming here as visitors or to work? Of course not. So it's hypocritically inconsistent, or you're just not thinking. That's my answer to it. And I want to be tempered, but I'm perplexed by the lack of movement here. It's not true where else we've talked to people, I can tell you.
Great. Well, good. I look forward to seeing results on that. You had mentioned something about the current vaccines that are in development for COVID-19, and you have some questions about that. I know work is happening at breakneck speed compared to how it usually goes with vaccine development. Is there a
You really believe
danger
that?
in that? What's your assessment?
Do you really believe that?
I don't know. What's your assessment?
I would say if you and I had the right money, as I said before, and you're sitting at your desk, in January 10th, Chinese published a sequence. It's an acute virus infection. This is not HIV. This is not that complicated. If you know it has an envelope or a spike protein sitting out there that's needed to attach to cells, and you have the sequence of it, and you have money, how long would it take you to make the RNA in mouse? And then you have to do it good laboratory practice and get it into people. It's 6 months since we've had the sequence, and that's breakneck speed. Well, we've got a lot of vaccine candidates coming forward, and the plan is, as I heard yesterday on those news conferences, we're going to look at 5 or 6 of them, and we're going to see how one may match the other, or do this better or do that better. Okay. If I've seen a tremendous amount of effort for an HIV vaccine go to neutralizing antibodies. And believe me, I was on that side. In fact, the first paper on neutralizing antibodies against HIV was Marjorie Robert Kurhoff and myself in Nature. I think it's 1984, at the beginning of 85, a long time ago. And certainly we wanted our vaccines to do exactly that. But I've never seen in a primate study when we got protection with HIV that it ever correlated with neutralizing antibodies. So it's not always the answer. But with an acute virus infection, there's reason to certainly to go in that direction. I'm not saying I would be against that. What I don't like is so much being that with lack of what is the plan if it doesn't work well, and who's making those decisions. Well, we heard that they're making a safety committee. Who are they? 1 or 2 people the same day? And they're making the evaluation committee the same day. I don't like that. I don't believe that will increase American ingenuity. And I pay a lot of attention to what's happening abroad as well. And I don't think anybody does in our government at all, or very minimally. When we have a global virus network call, we're talking to everybody, including Chinese, right? Including Russians. So I feel we need a little more, as I said over and over, plan B, a little more creativity, a little more thought. And I agree that that would be my first choice. I don't know if it would be my first, second, third, fourth, fifth, sixth choice, all going forward. And looking at this cross of how one may help with the other one, I mean, it almost looks like desperation to make sure some spike finally does something with a lot of help elsewhere. Hey, you know what? Wow. I mean, I don't know. I'm not happy with it, with that thinking. And you say, is there something that I would be worried about with the spike? Well, there's evidence in the literature, Chinese literature, that if you don't protect, antibodies to spike can make pulmonary disease worse. I don't know how. Is it forming immune complexes? Is it taking it to more tissue sites for infection? I don't know. I've also seen that durability can be a problem. If you look at papers from both China and from Holland with seasonal coronavirus, for example, less than 6 months. And there's evidence with this guy, not long lasting. At the same token, in recent months, we've seen evidence that if you're producing high levels of interferon, type 1 or type 2, you not only do better by controlling virus, you reduce inflammation. People forget that interferon also blocks overactivation of CD4 T cells when it wants to tell the immune system, okay, you've made your antibodies, now shut up. And in addition to that, when you therapeutically, when you give interferon, it's done the same thing. That's part of the innate immune system response. The next thing I say is, I don't know if it's true, because real secondhand, I have not verified this whatsoever, but I have heard that, we know the bats run around with a lot of coronaviruses and they don't harm the bat. I have heard it's a strong innate immune system that this is keeps in check. But this, I do know that this virus has expended some of its genetic information, and viruses don't like to get rid of any of their genetic information. They don't have so much to begin with to try to get away from innate immunity. It must have gone through an evolution period or a period of time when this thing was really knows to survive. It has to try to minimize this terrible onslaught by innate immunity. That's one of the things that tickled my thoughts about this and said, that is interesting, and became more quizzical about it. But it's the urgency of it all. Yes, I believe right now it could save life, and that's so it's hard for me to bumble around with it. Right? Okay.
All right. As you talk, I'm just aware that there are, at least from my understanding, a lot of kind of known unknowns about this virus. Can you talk about that? Where is the science as far as the understanding of this virus, and what do we know today that we didn't know, let's say 5 months ago? Is there anything that's concerning you that people aren't paying attention to?
Well, with one of my colleagues, David Zella, in collaborations with people in North Italy at the University of Trieste, we tried to follow the sequences and see if they were meaningful. The first observation that we made was that, boy, it's mutated a lot more than what they see in China, but it was so North Italy, much more mutations, the same as in the United States. So we saw that we have the same thing. I don't want to call it a problem, but much more mutation. What does that mean? We never found any correlation. That was months ago. There's some evidence now that some sequences might be correlated, especially when they hit the polymerase gene. That in turn makes it more mutable because that's the replication of the virus. And that might be associated with being less virulent. I don't follow it close enough to be expert to comment to a group like you might have. But the next thing I have seen I don't think it was literature, I think it was in some parascientific newsletter, that some things recently with some mutations look like maybe some things worse. That's what you're really looking for, right? I mean, it's nice to know that it's going out of business. Yeah, I'd really like to know that. I mean, I don't know how so many scientists prophesize, well, we're going to have around wave 2, we're going to have wave 3, et cetera, or we're not going to have any more waves. I basically say, how the hell do I know? I mean, it seems reasonable, but what happened? It would have sounded reasonable also with SARS, SARS-1, but we don't. So we're not yet sure about those things, but the sequence variability is something ongoing that I think needs to be watched, and that's where places of molecular biology power can make real contributions, I think, and then figuring out the biological correlations to collaborate with some biologists. We know where they are, we know the good ones, and who could measure this, that, or the other. I think that bridging is important for the future to try to make real basic scientific predictions. So we know a lot more about mutations now, but not yet sure that I can tell you, yeah, there's a strain, let's call it strain B, and that's doing this, that, and the other. That's still too early. But I think that might come maybe before summer ends, that we'll have that kind of information. what can I say? I told you what I thought might be a little new about the spike. I was impressed by this. I think it's Dutch paper with the seasonal coronaviruses that came out. Now, I only saw it about maybe 2 weeks ago. That, you know, that's less than 6 months, 4 or 5 months or so. That's really concerning. Yeah. So, I mean, that's important.
Well, let
Think-
me let me
think-
ask this. You talked about the polio vaccine as a plan B. As you talk, I just become more aware of what we don't know about this virus. In addition to pushing for that polio vaccine to get out there, are there any other things that you think should happen, should be in place as other plan Bs? So we just don't have just one plan
sure,
B.
I do. Yeah, yes. Actually, I would have made the oral polio plan A. You know,
Right.
I would have rushed it out right away, but when those days are gone, that's not going to happen. But sure, of course, we'd like to see. I personally, I'd like to see much more cellular T cell immunology basic going on in this, and hear from those people what they think are the best stimulants of T cell immunity. I would love to hear from an institute that might be, I wonder if there is one that studies aging. And it would be really nice to hear an answer. I actually raised this with Eric in talking to him. It seems nobody has asked that question. Maybe it's because the answer is obvious and I'm too dumb to figure it out. But in 1917, 1918, it was cytokine storm that killed. That's what we believe today, in addition to all the blood clots. But when they died in 1917, 18, they were young. Today, they're old. There's got to be a fundamental difference somewhere here, because it's the old that are dying like flies. The average age in northern Italy was 81, average. So every time somebody 75 died, you know, you got some older people dying. Now you can say there are a lot of old people in North Italy, and there are. But I think that's really a telling thing. And I would think that the Buck Institute should figure that out, not me.
I think, well, just to add, we are figuring that out. There's a lot of work here in collaboration with folks at Gladstone and UCSF and scientists around the world to take a look at the aging immune system, what happens as we age, and then specifically some of the drugs that Neven Krogan's group found show promise against the virus. They hit some aging pathways. So we're really drilling down and looking at the responses to those drugs and how we can put that to use.
I think it's terrific. So yeah, I didn't know your work, so therefore I thought I could joke with you, but you got me. Yeah, please inform me. I'm a well-qualified candidate.
I understand. Me too, actually.
Well, yeah, you got a way to go. I'm past the average age in Italy. You know, I'm 83. So this is a worrisome time. Every time I see somebody young coming in my backyard, I run.
Yeah, well, then I was going to ask you this later, but let me ask you this. What are you doing to keep safe? And what do you think about all the hoo-ha in this country about, you know, masks, no masks?
Yeah, I wear it certainly when I go outside, not in my yard, but when I go to store or things like that. My wife and I both wear masks. She's almost the same age, a year younger. Yeah, I try to keep a distance when I'm in the drugstore or elsewhere, best you can. And I mean, Maryland, I don't know if it's true everywhere. I mean, the aisles are one ways and you really don't get close to anybody. So I feel quite comfortable. Otherwise, look, it's the first time I've really seen spring since kindergarten, before kindergarten, right? We went to school all my life, right? Then I went to work. And when do we ever see the spring before? You can say, well, weekend, yeah, okay, a little bit. But, you know, to be outside every day, I've really seen my backyard. I know, talk to all the animals that are there. You know, I get to know them by name. So, you know, it's the different. So that part I hate to say it, I've come to kind of enjoy and realize you can do a lot at home. Of course, I can call somebody and say, how about making a presentation on my veranda with me 20 feet away, listening below my veranda? And I do that, and we have those kind of discussions. But I do a lot of, you know, email and conference calls with different scientists, not just in our institute, but in general. So you find that you really can accomplish a lot at home. All of us have learned that my friend at NCI, who's my neighbor, Jeff Schlom, is an immunotherapist. He says he never got more work done in his life than this period. Then but he also calls people over to his house.
Right.
So it's been different, but I have the positive side is also I have enjoyed seeing flowers open, leaves open. You know, I know what species are gone and declined since I really paid attention, and so on and so forth.
Got it.
And I'm sure you too.
Yep. So we have a question for you from one of our viewers. Do you think it is possible to detect characteristics of a new virus that we can predict it will be a pandemic pathogen? So can you talk about how do we, I think the question is, how do we stay ahead of this?
Yeah, again, you're not going to stay far ahead of it, and there will always be danger of a pandemic. If you look at the last 150 years, they've almost all been RNA viruses, and they come about every 30 years. You think about it, the flu of 1879, the flu of 1917, polio of 1950s early, AIDS of 1980s earlier, and now this, and in between some epidemics like SARS and MERS and some other things as well. In the midst of all of that was the great smallpox, which goes on and on and on until conquered. So we're always going to face that, and we're not going to, I don't care what somebody does. They can sequence every DNA on Earth. I mean, I knew there was the Virome project. They were sequencing everything. And then I saw a television program where they were in Africa, and there was an antelope, native antelope shot for food, and they were taking feces and saliva and blood and muscle, and with rapture and holiness, they were sending it to Cambridge as well as to Harvard, Cambridge, England, and Harvard for sequencing. And I thought to myself, you know, I didn't know how many viruses have ever come from an antelope, a man, but if we're going to spend money doing that, you better start biopsying my hamburger. At least I'm eating that, you know. I mean, it gets a little silly. So I don't think that I wouldn't put too much there. I like what we're doing with the Global Virus Network, virus centers of excellence as far and wide as you can, reporting on everything to a whole entire global group, and not bothering with government. We can all be together, whether Russian or Chinese, and the Russians have contact with Iranians, so I got to be a little careful with that. But by the way, the Iranians are starting an OPV trial of 130,000 people, and we're going to be aware of the data. I mean, again, I want to be a little careful of that one. So it's just, but it's now I had a senior moment. I lost the track of the main point I wanted to make.
That's okay. You were talking about epidemiology and efforts
predicting.
Find,
yes. What I'll tell you what the answer to that question I think is yes. I mean, if you did find a virus and a new disease coming, you could start making some reasonable predictions. Look, if it was a coronavirus, you have quite a bit of experience now. MERS epidemic, SARS epidemic, SARS-2 pandemic. So let's say another coronavirus comes in from bats and causes disease. I mean, you're going to get a little worried, right? I mean, that's for sure. But you have to be really stupid not to take that really seriously. But if I look more broadly, I mean, in my career, nobody really told me this or taught me this. It might be wrong thinking, but it's worked for me anyway. I tend to think of starting with a disease or starting with a virus. If you know the tropism of a virus, what cells it's going to go after, and if you understand the strategy today in molecular biology, the strategy for its replication, and you know a little bit about the immune response to it, you can kind of predict the disease and maybe even how rapidly it can move and spread. Conversely, if I see a disease and I think it's a viral origin, I start with thinking what virus, let's say it's a central nervous system disease, let's say it's starting to appear with increased frequency, let's say the country in Egypt or something, I'd start thinking of what are the classes of viruses I can go to the Global Virus Network that target the central nervous system disease, which experts do we have in that, what class did they fall in, who are the best in the world on that, and what do we make the kind of virus this should be. And it's replicating very slowly, it looks like, from the epidemiology. What would that tell us, and moves like that. So there are things we can do at a basic level, but we always need it combined with public health experts and epidemiology people.
So a question just popped up. Is there a reason to expect pandemics more or less often than every 30 years? Because you mentioned that 30-year window.
Well, they've often dreamed of being God, but it didn't work. You know, what do I know about that? I mean, if somebody gives you an answer to that, you know, they are being hyper speculative. I can be hyper speculative too, but we don't know the answer to that.
Got it.
We do know. If I was talking to the public, and I'm not, I'm talking to you guys.
Well, you're talking to, there's public listening to you now.
Well, I have one word for what I always say when think change. One word, change. Change of virus, yeah. Polio. It used to be probably a Coxsackie enterovirus that was in your gut and doing nothing, but it became neurotropic. Not good news. Or society change. Think about it. Refrigeration and air conditioning, Legionnaire's disease. The forest is smaller. In my backyard, I live next to Congressional Forest. Lovely area with a river going by. However, the deer have less room. They're in my backyard. The deer have ticks, Lyme disease. The crows in my backyard carry West Nile these days because of global warming, right? Other things like insects came a little further north, like Zika, change, warming. Or the Romans opening up a new port in the Eastern Mediterranean. New epidemics came to Western Europe and to the Eastern Mediterranean. Columbus came to America. He gave a gift to the Indians, tuberculosis, smallpox. The Indians returned the favor with syphilis brought back to Europe. So what happened with HIV? You know what happened. It used to be in the rainforest and limited. And then the colonial powers left. People came to the big cities, Kinshasa. Prostitution went up because poverty went up. It became more common. And how did it become global? How do we think? What changed after World War II? Travel, tourism, money, availability. The sexual promiscuity came on strong. And the insanity of global intravenous drug abuse. That explains the spread of HIV, change in society. So every time, that's the way you have to think about it. So will there be another, of course there'll be another epidemic or a pandemic. The questioner knows that, and I'm sure maybe better than I do. But I could never say it'll be more or less frequent. I can say, if we keep making more changes, I don't know what happens if, let's say, the Amazon rainforest continues to get less and less. In global warming continues and more mosquitoes move further north. We can certainly predict Zika moving up further north as it gets warmer, as the mosquitoes migrate. Another change is boats, ships, whatever you call them, going all around with cruises, right? So you had much more norovirus epidemics, not pandemics, but also rats, mice, they travel. So do mosquitoes on airplanes and boats. Airplanes are another big way things are spreading. So would you anticipate that there will be more? Yes. If I'm a betting man, yes. Would I expect it a little more frequently? Well, if you look at the last 50 years, they seem to be more frequent. And the way we're becoming a smaller globe, yes, I expect it, but I sure don't know it.
So I'm getting some questions here about young people, children. One question wonders, it sounds like the vaccines that the young people are getting, either, for mumps, measles, rubella, might be keeping them resistant to COVID-19. Do you think that's a possibility? And then there's also a question about if you have any insight about why there are some young people who are having pretty serious reactions to this disease. So it kind of goes both ways.
Yeah, but very few young people have serious reaction. At one time, Bob Redfield, CDC director, a friend of mine, and my former colleague, he didn't think anybody under 30. He was really, we were talking about experimenting with SARS-2 itself, going into young volunteers. Then quickly, I got a call from, oh, Jesus, forget about it. Just got so-and-so and so-and-so data coming in. But nonetheless, come on, when the average age in Northern Italy, where there's a tremendous epidemic, is 81. That tells the whole story. And I mean, how did Italy get hit so hard? We know that the first virus that entered Italy is from Frankfurt, Germany, the airport, undoubtedly. But Italy's fashion industry around Milan is a tremendous number of Chinese people learning fashion there. I heard it's like 220,000 people. That's big for Northern Italy, right? And a lot went home for the New Year and then came back. I suppose that had something to do with things starting and really pushing there.
So back to the question, do you think the fact that young people get regular vaccinations, do you
Yes,
think,
yes,
that
yes.
could be behind
could.
some of the,
Yeah, it could, but I don't want to, it's a hazard if I say yeah, because then they'll say, oh, look, he's using data that doesn't exist. So I can't say, but would it make sense? Yes. Could. And we don't know how long this thing lasts. So, but if somebody got vaccinated, let's say a 6-year-old or a 10-year-old, it might give them a year of protection, maybe longer. If you believe the BCG molecular imprinting, maybe it can last significantly longer. But I don't want to claim that, but it's not irrational. It's the thought is, of course, occurred to us, but we've never said any, we don't bring it up at all because we just don't know. And I don't have the statistics. Well, yes, this is hard. I don't get infected with this virus very much, but when they get infected, they don't get disease much. That's another thing we don't control, right? So that could be the reason. Not that they were protected, but that they got infected and they blew it away. Maybe because they have a much better innate immune system at that age, right? Also.
right?
Yeah.
So that brings me to a question about where we are with testing for this disease, both who has it and also antibody testing. I mean, people are looking at, antibody testing as that'll be really valuable. Do you think we're going to get there, and will it be valuable?
Well, first of all, yes, I do believe it'll be valuable. Yes, I think we will get there. I think we're already there. I think there are good tests now coming into play. It took a long time. Bob Redfield took the blame. If you know the story, it's a little sad. I mean, CDC apparently had a pretty good test, and then they were informed this is the, let's call it for the sake of simplicity, I don't know what it was. It's a reagent, let's say nutrient broth. That screwed the test up a lot. My colleagues and I developed a blood test for HIV, both the first and the second generation tests. And people would say, well, gee, that was really fast and really good and very specific and all that. Yeah, but there were some things better about it. We could relatively safely mass produce the virus. We were not afraid of it. We just didn't use glassware and needles. So we had a lot of material to work with. And then we collaborated with people in Frederick, Maryland that had these enormous vats. So we had loads of virus to work with. That's one. And so we didn't have all these fears of things. Number two is that the antibodies to HIV proteins, did not cross-react with the retroviruses, human retroviruses that we had discovered just before, known as HTLV1 and HTLV2. Those viruses are human retroviruses. One can cause serious disease, but it didn't cause us any problem with testing because there's no significant cross-reaction. Whereas with this SARS-2 problem, there is cross-reaction with the seasonal coronaviruses, for example, some creating a little problem. So one of the key things, and people are there now, I'm sure, was to focus on the segments of viral proteins that don't cross-react, to find those epitopes and really produce them and really make better tests. And I think that's probably developed. I keep saying probably because I don't have it in hand. I'm not sure what company. You'll ask me specifics. I know a lot of people are working in that line, and I know the tests are getting better.
And do you think that they will be a key factor in tracking this thing?
Yes, but what worries me more, was about to finish, is the false negatives. I mean, that's really scary because you could say, well, those people didn't make antibodies. Well, maybe they did, and the antibodies have already gone away. What I was worried about before, the spike antibodies, maybe they're gone. Because that's a strong antigen, and they're gone. I mean, so that is one. They say, well, those people didn't make antibodies. Well, are we sure of that? Did we look early? Have we followed people from early and see how long the antibodies last? That's being done now. I suspect these things are not lasting a long time, and that's a worry. Both for the immunity to the people, for the idea of a herd immunity, and certainly for a vaccine.
So I listened to parts of the hearings yesterday, and when they were talking about vaccine safety, there was some comment about a concern that if somebody had a low response to the vaccine, that they could actually get a worse case of COVID-19 if they happen to be infected. Are there concerns, or what's happening with that, or what could
Well,
happen?
there are reports in the literature from China at the very beginning of exactly that. That's what, look, if you talk to outstanding world leaders in coronavirology, we have some in America. I haven't talked with them, Barbic in Carolina, for example. But Ab Osterhaus from Holland, now in Hanover, Germany, or Johan Netz in Leuven, Belgium, you know, they insist on animal model testing, and they're working with several animal models that they developed. Yesterday, I heard it said in that same hearing, when we just developed animal models. Well, we didn't. They were developed earlier in Europe. I don't know who we is, but you know, that's gets sometimes a little much. But they worry about that a lot. Those virologists, that we could get some kind of enhancing antibodies that really don't simply enhance. They may be making immune complexes. They may be distributing the virus differently. One Chinese paper said they got worse pulmonary disease. I don't think the mechanisms are understood yet. And I don't think everybody has crossed that bridge to accept that. But certainly there are papers reporting something like that. And certainly the European continental colleagues that I talked with on those telephone calls that I told you about push for these animal models being carefully looked at before rushing to vaccines in men.
So what, when a vaccine or vaccines become available, what needs to happen for you to feel comfortable getting a dose yourself or having your loved ones get a dose of a vaccine?
Watching what happens with other people and listening and talking to my GVN colleagues that I trust. And I mean, you know, let's see what happens when they first have you've got a lot of volunteers, and I don't, you know, I don't expect any toxicity. But when they talk about toxicity, they're talking about acute toxicity from the vaccine per se. This takes time to determine what the toxicity is, like things you're talking about. That'll take a year or two. I don't know.
So do you think there's some hope that we could have a vaccine by the winter? Do you think that's reasonable, or do you think what, you know, I mean, I know, and it's a bet. I understand if you had a window of when you think a safe, effective vaccine could be available, what's your take on that?
Well, I think we've got to make a promise. They won't do it on the nightly television stuff, the government people. We have to promise we will say vaccine candidate. Because once again, you have a vaccine when you have a vaccine. That takes time to evaluate. So if you're saying by winter we're going to know all this, of course not. And you know, and that'll already be, since it was first announced that it would be 5 months or 11 months, that'll already be about that time. Way past 5, and it'll be then. We could know acute toxicity, right? We'll know that. We might even get some efficacy data by late fall. That's possible. But we won't know the full impact of safety and durability and future help to second waves, et cetera, before 1.5 years, maybe. From when? Maybe even from now, but another, from the time you have it, probably another year is my best guess.
Got it. So I want to talk to you about the Global Virus Network, but before we get to that, what about our relationship with the World Health Organization? How important are they in all of this?
Well, I mean, I have pros and cons about, you know, we need WHO and we need to be part of it. So let me just make that as the conclusion. I mean, they have good people. If they have an unknown, like HIV, the experience wasn't great because when it's an unknown, they don't know who to call, right? And they sometimes have directors, general directors. I don't want to go through past history of negativism, but they've had a past director that didn't think we should do anything with HIV, met with me privately Remember, I said I discovered the first retrovirus, that's HTLV1. It causes leukemia. It's endemic in certain countries, not in America or in Europe, but in Japan, for example. This man was from Japan.
Hmm.
And he said, Bob, you and I know it's much more important HTLV than HIV. I almost dropped because HIV was going like that. HTLV is an ancient virus, and it is where it is. We know where it is. It's important for the people in endemic areas. It's very important. And if you get a blood transfusion from the wrong person, it's going to be very important for you. Or have a partner that's the wrong person, it's going to be important for you. But it's so much more difficult to transmit. It was such a less urgent problem. So is that WHO in general? No, but it can happen, right? So they have to be, by very nature, political, because they represent all the nations. So that's a very sensitive position for them. I don't envy it, but we must be part of it, and we must be supportive of it. The last thing we need in a global epidemic, i.e. pandemic, is to be nationalistic and to be with ourselves only. That's why I love the GVM.
Right.
Because I know I get total story, not just what is in the interest of one person or in the interest of the president or this, that, and the other. You know what I mean? You're talking to everybody.
So we got a question, and we haven't talked about antivirals yet.
Mhm.
And this question is, and it's specific, but you can also talk in generalities, obviously. If someone is taking a daily antiviral, like Valtrex, which is used for herpes, could it provide some protection? So there are antivirals out there. Feel free to say you don't know if you don't know, but what's your take on that? People are so desperate.
I understand. Of course, that's desperate. But antivirals, what does it mean? You got to be specific. Viruses differ, like you and I differ, just as much. I mean, they're different one from another. You can't throw one thing for this to that or the other. And so, but that doesn't mean that Valtrex wouldn't be helpful. I cannot say that. I can only say I don't know. But you certainly can't say because it worked for this, it should work for that. That's no way, right? When do you think antiviral efficacy and real effect first happened historically? When do you think it was? Do you have? I'm asking Christine Rebelo.
I do not know.
Well, I want to tell you something I believe is historical. I believe the first time a systemic, the words are important, serious virus disease was shown objectively, so those are the important words that I emphasized, to be treatable was in 1985. It was in my lab, and it was principally by Sam Broder. We made a visit to Burroughs Wellcome at the invitation of Duke University. We went there first. Sam Broder decided to come with me. He was a clinical guy doing clinical research. And he talked with a guy named David Berry at Burroughs Wellcome. They had a shelf load of compounds that were made for cancer just sitting there, realizing that they could be polymerase inhibitors and that reverse transcriptase is a DNA polymerase. Broder wanted to push testing them. I was a little scared because I said, people with this disease, you can end up killing them with this kind of drugs, I'm afraid. But we published the first paper, I forget, it might have been Lancet, and on in vitro studies. So we showed we could inhibit the enzyme of HIV, reverse transcriptase, better than the DNA polymerases of human cells, which we happened to be working on in my lab at the time also. And then in vitro, you could block HIV with AZT without so much toxicity to the cell. Now that was the end of it for me. Broder did this in my lab because he didn't have a virus laboratory, and so we set it up for him and with our culture systems. But then he wanted to go right to the clinic. I said, "Oh, here's where I say thank you very much." I was afraid. I really was afraid that we were going to really cause harm. He made a major impact with a major paper. He and Bob Yarshon, who's still at NCI, National Cancer Institute, where this was done. And that was showed virus going down, immune system coming back objectively, patients doing temporarily better. Wasn't the answer, but it was the beginning of the beginning. And it brought the pharmaceutical industry. And by 10 years, you had all the anti-HIV therapy you needed. And better and better and better. That's the story. So now you want to just take any drug that we use for HIV and dump it on here? It doesn't work quite like that. You've got to have a specific, why do we have antiviral therapy at all? Why didn't we have it earlier? Because, viruses don't have they're not alive. They're inanimate. I mean, they use us to replicate with a few genes of their own. So you have to have a clear understanding of the molecular biology of their cycle of replication. That came from molecular biology, from basic research, the kind of things that are done at your place. So by 1984, 1985, we had the ability to see the stages of HIV replication in detail. We knew it in our lab very, very well at the time, and almost completely. I mean, most of the things that are targeted today were known then. And so you could much more laser sharp attack. And that's changed the whole world, that you can now have somebody ask a question, well, antiviral therapy. Well, that was just for animal studies at the time. I remember doing reverse transcriptase inhibitors for my studies in mouse leukemia, but you wouldn't be, what are you doing for that for human? No. So that's the answer I can give. I can't say no, but I sure can't say yes. And if it's got toxic side effects, be really careful.
So we're getting close to the end of our time. This hour has sped by. Talk about the Global Virus Network. What is it doing in relationship to COVID-19?
I'll do my best. But to close the other, I just wanted to say, we should have more drugs than remdesivir, which you get out of hospital a few days earlier. And that's what was talked about yesterday. After 6 months and a lot of money. I mean, come on, we've got the world working on this. This is a discipline so far. So, and I wish that question came up in the hearings. So for the GVN, it was a concept that came to me out of what happened in HIV. I used to hear from the late Jonathan Mann of WHO, this was the fastest pace in the history of medicine. From a mysterious, unknown disease where you couldn't find anything, and blah, 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 becoming a global pandemic, et cetera, to having the gene sequences, the blood test, the cause, blah, 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 everything you wanted. And then I thought, we could have really done better. And the things that happened in laboratories happened by chance, because the CDC would have had some responsibility, but they didn't know about retroviruses. NIH, well, it was the Cancer Institute, Allergy Infectious Diseases, where our friend Tony is, they had nothing to do with retrovirology, minimal, almost nothing. So, I only got involved by chance, as anybody else that was in that early period. I happened to hear a lecture from James Kern at CDC, who was the town crier. He really made people aware. And he made a talk at NIH, and he said, where are the virologists? And there were like 6 patients in New York. And I said, we're not going to change fields I was working in cancer before that. So I had 2 careers, the NIH came. But he came a second time about a month or 2 later. And when he brought up the new statistics, I said, he's right. And we work in T cells, and we know how to grow T cells. And this damn thing sounds like the viruses that we knew that were retroviruses. Okay. So, they blood, sex, mother to child was transmitting it, targeting CD4 T cells. That sounded like a relative of HTLV1. The irony is it was a retrovirus, but it's not a brother of HTLV1, it's a cousin. So that idea spawned in me that we really need to be sure there are enough virologists, particularly when I was told by people at the Karolinska Institute in Sweden that they too hadn't seen a new virologist, a young man training in, or a woman training in virology. The youngest one was 55 years old. And I said, this is all up to chance. So we need to get a group of people that have the best experts we can find that cover every virus that's of danger to man, and have centers of excellence connected as best we can. The president is Christian Brichot. We recruited him a few years ago. He used to be president of Pasteur Institute. And it's working. I mean, we have a course in the summer. We have a meeting every year that I don't know what we're going to do this year for the course or for the other part, but we got to work something out. And we're making more young people aware of virology for all these years that this has formed since 2011. Its goal, train more virologists, let the public be available to be advisors to the public, speak forthrightly when asked questions by Christine Rebel, and to the Buck Institute, no political pulling punches, tell what you really believe, advise public health people when they ask, try to survive without governments by begging for money from the Buck Institute, et cetera. You know, I'm a terrible joker like that. But you know, it's that those are true things. We have to hustle for getting a little money, but it's working. And we also, other than training virology, giving advice, we do have things that respond. You wanted about the pandemic, and yes,
I did. And
yes, the
but
oral
before-
polio vaccine came out of those discussions, for
Great.
example. Oh, yeah, and
So-
we're doing other things in serology and helping and really trying to work to hear the latest in people in the GVN that are developing drugs, really developing drugs, and really doing the work that's developing animal models. And we're making those things available as fast as we can. And we hear everything that China knows from China directly. Okay.
So before we go, my last question, is there anything that I haven't asked you, or here's your chance? Is there a final message to the folks that are watching today that you think they should pay attention to or that they should care about?
I can be serious and close with a little, like I've been doing. You should get more than one source of your information. It is not always just coming from the nightly news. Keep that in mind. It's important. Look more broadly. Look to your fellow scientists in your own region and your medical health people in your own region for opinions, firsthand opinions, and to the medical centers and to the institutes. I mean that genuinely, not as patronizing this institute. Don't just rely on, you think, well, that's somebody that knows everything. That's not exactly true. You got to have more than one source of information. I think that's important. Be aware of the Global Virus Network. We would not be unresponsive. And we do have information flow from abroad more. And the other side is support the Buck Institute and make them work faster and harder, because I'm 83 years old. You know, it's in it goes faster and faster, and we want them to understand the intricacies of the aging process in immunology.
That is great. Thank you. That was an excellent way to end it as far as the Buck is concerned. Thank you so much. Thanks for taking time with us today. And I also want to say thank you to everyone who participated in today's webinar. A video of today's event will be posted on our extended session website later in the day or tomorrow. Our next session happens next Monday, June 29th at 11 a.m. Pacific, 2 p.m. Eastern. We'll kick off the week in conversation with Dr. Werner Greene. He is a distinguished professor of translational medicine at the Gladstone Institute of Virology and Immunology. Dr. Greene will be the perfect person to update us on the latest state of the science when it comes to COVID-19. His lab is studying how the SARS-CoV-2 virus enters cells and is screening a library of FDA-approved drugs to identify those that could be repurposed as a treatment to stop cellular entry and the spread of the virus. He is collaborating on many important projects. There will be a lot to talk about. So please enjoy the upcoming weekend. Stay safe and healthy. I will be back here on Monday, and I hope you will be too. I'll see you then.